Good afternoon, everyone, and thank you so much for attending this webinar session. Welcome for those of you who are not members of the IMechE to the Institution of Mechanical Engineers. It's lovely to have you here. We as an institution are a membership organisation of engineers from across a range of engineering subject matter areas and sectors, which I'll go into in a little while. But today's webinar is on pulse oximetry and particularly focusing on accuracy bias and the impact of patient care. We have some incredible speakers for you today, and I'm very excited to have them here. A mixture of engineers, scientists, clinicians, who are really going to tell you about the issues surrounding this problem. For my part, I'm really just the host here today. So if you have any issues, myself and the team at IMechE, who are backstage right now, will be able to help you if you're having problems with Zoom or anything like that. But I do hope you enjoy today's session. I'm the immediate past chair of what is the Biomedical Engineering Division of the IMechE. And one of my roles within that is to host these kind of events. So we're very pleased to have you. By way of just giving you an idea about the institution has been around since 1847. It is made up of engineering professionals from around the world. It is not just a UK organisation, and it is based at One Birdcage Walk in London. We have members across the world, as I said, and covering a wide range of engineering subjects. Our main focus, we are a charitable organisation as well as being a membership body, and our main focus is to campaign and promoting engineering to wider society. And in that vein, today's presentation is quite meaningful. As I've already mentioned, we are made up of a significant number of engineering fields, but today's event is really being hosted by the Biomedical Engineering Division, which covers everything from medicine, healthcare, as well as medical devices and technology. But as you can see, we cover a whole range of other areas as well. So please feel free to join us for any of our webinars. They are all free. They're all usually this time in the day or around lunchtime, and you are always welcome to attend if you're interested in any of our subjects. We have about 115,000 members worldwide, but the division itself is made up of about 3,000 members. These are people who are working in industry, academia, and in healthcare as well. And so on their behalf, I thank you for attending today. I'm going to just close by saying, if you do want to get in touch with the IMechE, if you are an engineer by profession and are interested in joining the institution, then please do reach out to us. But with that said, I would like to introduce our first speaker, and I suppose the co-host really of today's event, and that's Nick Gompets.
Great. Thank you very much, Helen. Really pleased to be here, and thank you very much for hosting this. It's amazing that you've taken this on board. What I'll do is just share a few slides, say who I am, why I'm here doing this, talk about what the webinar is going to be about, and I'll structure it. And one thing just before I forget to ask you is that we can't see who you are. So if you're willing to introduce yourself on the question and answer slot, that would be fantastic, because I hope we've got clinicians and industry members, regulators, guideline writers, clinicians, and obviously biomedical engineers. So I hope we've got a whole range of people who can actually comment on this. So really this is to raise awareness and start a conversation. So I was an NHS doctor for 30 years. I started off doing hospital medicine, got my membership of the Royal College of Physicians, so I was a medical registrar for a bit. So before pulse oximeters were available to most of us, and they were just confined to anaesthesia and intensive care units, I was putting needles into people's arteries to measure their oxygen levels. But since then, I've experienced how pulse oximeters have come into primary care as a primary care doctor or GP. So really, I was slightly concerned when I realised some of the accuracy issues regarding pulse oximetry, and that's why I wanted to discuss this. My conflict of interest is I gave up general practice 4 years ago and now founder of a medtech business where we're trying to improve pulse oximetry. But that's all I'll say about that at the moment. This is about general pulse oximetry, not about anything we're doing. So just to move on to how the structure is going to be, you've heard from Helen, thank you very much. We may be hearing from Paul Silverstone next. We've had a bit of a technical issue. He's a professor of general practice and wrote a BMJ article in 2022 on training about oximetry, and will give the overall view of pulse oximetry use in primary care. To Dr. Zaifa Adamali, who's a respiratory consultant or pulmonologist, and he's going to look at the use of oximetry and the importance of accuracy in hospital care. Sam Morland, who's a technologist, a medical devices developer, and we'll look at sort of device development and technology limitations. And finally, back to me. Unfortunately, I managed to ask for a date that happened to be Thanksgiving. So although we tried to move the time so it's appropriate for America, I think if you've joined us from America, happy Thanksgiving. I'm sorry to interrupt your day. For those who listen to us later, then I hope you had a good meal. So unfortunately, I'll be presenting the open oximetry, which has come out of the US. And then really it's less of questions and answers. It's more about comments and opinions. These opinions we are promoting here are our own opinions. We have very similar opinions, but not necessarily completely aligned. And there are obviously other opinions that are probably maybe counter to ours. So certainly interested to hear regulators, industry, etc. So if I may pass on now to whoever is next on the agenda, just to say Sam Morland has had a new baby recently, so he will be presenting by video to begin with, recorded, and then come hopefully to the question and answers. So thank you.
Greetings and welcome, everybody. Sorry that we've had a few little technical issues, but hopefully we've got over them. So greetings and welcome. Good afternoon to you all. My name's Paul Silverstone. I'm a visiting professor at Anglia Ruskin University and visiting professor of primary care at the University of Suffolk. And Nick has very kindly asked me to talk about pulse oximeter accuracy and especially how it affects patients in primary care. So let's step back a second and just think about pulse oximeter accuracy. So when you take a pulse oximeter reading, pulse oximetry is about measuring the amount of oxygen that is in the bloodstream that is attached to hemoglobin. And I make that point because pulse oximetry is different from looking at an arterial blood gas, which involves sticking a needle in someone's artery and taking some blood from it, and looking at what is happening at that end. So it's important to think about accuracy because when we're talking about it, we're comparing a level of oxygen in different places, if you like. So that's why when we compare pulse oximetry readings to blood gases, we're looking at something that is slightly different. And that's the first thing that we need to take on board when we're thinking about accuracy, that actually what we are looking at is slightly different depending on where we are analysing, in effect, that blood oxygen level. And that is why when you look at the FDA and MHRA warnings that were issued in 2021, they repeatedly make the same point, that pulse oximetry readings are only an estimate of the gold standard arterial blood gas, because one is measuring oxygen on its way and the other one is measuring the amount that oxygen is being delivered. So if you think about it, it's a bit like an Amazon tracking order. So pulse oximetry represents that tracking order. It's on its way, but it doesn't tell you whether it's actually been delivered or thrown over the fence or been delivered to the wrong address or whether it's any good when it gets there. So it's important to think about that when you're talking about accuracy, because you could have a device that is absolutely accurate, a pulse oximeter that is accurate, but it's not telling you what is happening downstream where the cells are, which is where the oxygen is actually being used. So we always need to bear that in mind, and that is why the FDA and MHRA make that point. When we look at accuracy of pulse oximeters, then we need to think about the different factors that can affect accuracy. So we've got the equipment itself, the hardware and the software. We've got the way those devices are tested and how their accuracy is reported, which is what Nick is going to talk about in a minute. But it's also about other things like how those devices are maintained and how they're calibrated. And there was a paper that came out a few years ago, which found that 1 in 10 hospital pulse oximeters had a fault with it, often cable faults. But there it shows that we've got to consider all of these things, just thinking about the equipment factors. So even if we get a really good device, we need to make sure that these other issues are discussed as well. We need to think about patient factors, because the way that these devices work is looking at hemoglobin and the amount of oxygen and carbon dioxide that is attached to hemoglobin. So if you don't have a lot of hemoglobin going round, your device may say everything is okay because there's lots of oxygen on that one remaining molecule of hemoglobin. But actually downstream, the cells are not getting enough oxygen because you haven't got enough hemoglobin. Or maybe it's the quality of your hemoglobin that's not very good, that you've got an abnormality with your hemoglobin or something that is attaching itself to hemoglobin and stopping oxygen being attached. You have problems with light absorption. So if too much light gets in under the probe site, that can mess with readings. And equally, if you've got something blocking light absorption, so it could be something like something on the skin or something under the nail or the color of the skin or the thickness of the skin, that affects light absorption and give you an inaccurate reading. Or because it's looking for pulsatile hemoglobin, you could be having a problem that there's finger movement, which mimics pulsatile hemoglobin, or the other end of the scale, you're not seeing enough movement because you are having problems with perfusion at the site that you are doing the measurement. So a patient who is cold, a patient who is shut down, who is septic, or who is shocked. And you have problems in the downloading of hemoglobin, oxygen from hemoglobin, that can mean that your pulse oximetry reading is not picking up what is going on downstream and therefore not accurate in terms of what is actually being delivered. Then you have sampling factors. So you could have a brilliantly accurate piece of equipment, but if you don't prepare the site where you're going to put the sensor, so if that site isn't warm, if it isn't clean, if it isn't held at the right level, then you may get an inaccuracy. Or you have a problem with signal quantity. So if your finger is cold, for example, or signal quality with the finger being waggled around, or the exact timing, which Sam is going to talk about. So if you are doing a single point in time reading, exactly when you do that reading can change from almost second to second. So we need to think about these causes of inaccuracy, and we need to think about some other factors like the size of the inaccuracy. Are we talking about a tiny little bit of inaccuracy that has no clinical impact, or a large inaccuracy that could? Are we talking about something that happens very frequently or something that happens very infrequently? So, for example, one of the things that caught us out during the pandemic was that things that happened infrequently in normal clinical practice became a major problem because of the frequency with which it happened when you're dealing with large numbers of patients. Are you dealing with an inaccuracy that affects all patients equally, or specific groups, and do we know who those groups are? And what are the clinical consequences of the inaccuracy? An inaccuracy doesn't matter if it doesn't have clinical consequences. So what are the clinical consequences? Are they serious? And the final part of this is, what is your clinician awareness of the inaccuracy and the causes of inaccuracy and the consequences of it? Because if you have people who are not aware of these problems, that just exacerbates the situation even further. So bearing that in mind, let's just look at that and apply it within primary care. And what I'm going to do in the next few minutes is explain to you why accuracy matters in primary care. So when a patient comes in to the surgery, or when you go and visit a patient at home, primary care medicine is really very fundamental. So we're wanting to do three things. We're wanting to assess the patient. How sick is he? And what's making him sick? We want to know about the management of that patient. Should I start treatment, and with what? And we need to make a decision at the end of that consultation. Should I refer this patient to hospital? Should I review them again and see what's happening, see whether things have got better or worse? Or should I send them home with safety netting advice to call us again if things get worse? So primary care medicine is all about making very basic, fundamental decisions. And we're using very basic tools that haven't changed over 200 years, other than the fact that we've got a stethoscope in the ear instead of a rolled up piece of paper or a shirt on someone's back. But these tools are very basic, and they have limited specificity and limited sensitivity. So we are trying to make very important decisions based on some very basic tools. And that's why pulse oximetry, when it came along, was so valuable to us. It is a valuable tool because what we can do is add that to those basic tools and throw some science at this and throw some numbers at this assessment and management and decision-making process. And that's what makes pulse oximetry so valuable in primary care, because what we can do is we can put it on a patient's finger and it gives us a feel for how sick he is, which means we can triage patients. And we triage patients in a fairly sort of straightforward way, in a way of traffic lights. So we look and see whether our patient is big sick at one end of the spectrum of sickness, little sick at the other end of the spectrum of sickness, or somewhere in between. And we can then say, right, the patient who's big sick needs to go to hospital, the patient who's little sick can go home, and the patient who's somewhere in between, we might review them again and see what's going on. Okay, so it then allows us to triage patients. But I want to point two things out to you. Firstly, look at the numbers that we are using. They're numbers really based in that area between 94 and 100%. So it's a narrow range of numbers. And the numbers that we are using here have very narrow thresholds and cutoffs between determining whether someone is big sick or little sick. And that is important when you apply accuracy to this. And similarly, we follow assessment guidelines, which Nick again will talk about in a minute. And those guidelines, again, feature either absolute numbers or very narrow thresholds. So that means, again, that accuracy becomes very important when you follow these scoring systems. And it allows us to identify what the cause is. So if we've got a patient who is unwell, It helps us in our assessment because we can spot things. So if someone has passed out, by putting the pulse oximeter on the finger of that patient, you could pick up whether they've had a pulmonary embolus, even if there are no other signs of a blood clot having travelled to the lungs. The fact that they are short of oxygen is an indicator that that might be the cause. Or a patient with a high fever, the fact that they are short of oxygen could indicate that they have an atypical pneumonia that isn't showing other signs. Or a child where it's quite often difficult to spot the symptoms and signs of pneumonia because they don't present like adults. The ability to put a pulse oximeter on and see whether that patient has a low oxygen level is really helpful. It allows us to pick up these different causes, which nothing else would help us to do. And it allows us to follow management guidelines. So if I want to know whether to give this patient oxygen, I can follow a guideline which tells me whether they should or shouldn't be given oxygen. But again, I draw your attention to the point that they are using sort of single numbers, absolute numbers, or very narrow ranges of numbers within guidelines for this decision making. And whether the patient is sent to hospital or is reviewed again or is sent home with safety netting advice, particularly during the COVID pandemic. Again, it allows us to follow a guideline in which pulse oximetry readings are incorporated and decisions are made based on those readings. But again, if you have a look in the bottom right-hand corner, they are very narrow ranges or absolute numbers. And again, that places the emphasis on accuracy. And therefore, it's important what the clinician's perception of pulse oximetry is. Now, primary care clinicians consider pulse oximeters to be reliable and accurate because they're unaware of the factors that could change that perception. They perceive that pulse oximetry can rapidly and reliably confirm or exclude hypoxemia, can quantify the precise degree of hypoxemia, can facilitate the triage of patients into big sick, little sick, guide the management of patients, give them oxygen or not, and be used to base critical decisions on sending them home or sending them to hospital. That is the perception of pulse oximetry by primary care clinicians. And in many ways, pulse oximeters represent a perfect tool. The patient comes in, you put it on the finger, and you continue asking the patient questions. It's not like doing a blood pressure reading where you actually have to stop the consultation and do the reading. You're actually just putting this on the finger, continuing with the consultation and having a look at the reading in a few seconds' time to see what it is. It's the ideal tool in primary care. It's easy to use. It gives you a rapid integrated assessment so that you're talking to the patient and doing a reading. You don't have to stop and count a respiratory rate and a heart rate and so on and so forth. It's simple to read. It gives you numbers, unlike an ECG, which you have to interpret. And the readings are considered to be reliable and accurate. It's an exact number that it gives you. And more than anything, you just take it out of the box and use it. No training is required, either in using it or interpreting or evaluating the results. And above all else, it's cheap. So it is the perfect tool for use in primary care. But the problem is it's not a perfect tool. It is a valuable tool, but it's not a perfect tool. And it becomes less of a perfect tool because of the types of devices that we're using, which often are not of the best and most accurate type. You find that clinicians are using a one-size-fits-all approach with pulse oximeters. So you see them using adult pulse oximeters in children. And, when you talk to them, they say, oh, well, on small fingers, I turn the pulse oximeter sensor around at 90 degrees. because it won't fit on the finger otherwise, or I put it onto the earlobe. I've heard both of those in the past month. Okay, so we know that we are having problems with equipment. We know that we have patients with all cold hands, with darker skin tones, with all sorts of things that can affect accuracy. So we've got a wide range of patients with causes that might cause inaccuracy. And we know that there isn't a standardized approach to doing a reading in primary care. So patients are wiggling their fingers around and people are talking to the patient at the same time. And there's all sorts of things going on which could affect the accuracy of a reading in primary care. And therefore, what you have is a perfect storm here. Okay, you've got equipment inaccuracy, you've got patient factor inaccuracy, and you've got sampling factor inaccuracy. And you have clinicians who are untrained and unaware how to use pulse oximeters correctly. They're unaware of the inaccuracies that Sam and others are going to be talking about, and they're unaware of the factors that can cause inaccuracy. So they don't even know which patients have an increased risk of inaccuracy. And they've been untrained and they're unaware of how to interpret and evaluate pulse oximetry readings correctly. So you can see here that there should be some alarm bells ringing, okay? And why this matters is this, that we are making assessment, management, and critical decisions based on guidelines that feature either absolute numbers or a narrow range of numbers. So when you factor in inaccuracy, unawareness, and guidelines that feature very narrow ranges or absolute numbers, you have an even bigger problem, because even a 1% level of inaccuracy in that situation has the potential to result in serious harm to patients. And you think about it here on the right, manufacturers talk about 1 to 2% level of inaccuracy in their devices. The FDA and MHRA regulators talk about between 2 and 4%. Then there are those equipment factors, patient factors, and sampling factors, all to add in. So that reading of 95% could be anywhere, okay? But we're making assessment, management, and critical decisions based on that reading being 95% and applying that to a guideline where 95% matters. And this means that we have real-world implications from pulse oximeter inaccuracy. Patients get wrongly triaged. They get a wrong illness severity score because illness severity scores depend on very narrow ranges of numbers. Hypoxemia gets missed because if you have a reading that is more than 5 or 6% out, even a reading of 95% could mean that the patient is actually hypoxic. A patient might not be given oxygen because the pulse oximeter reads 95%, but it could easily be 93% when they would need to be given oxygen, or they don't get sent to hospital, which is what happened during the pandemic, because people were falsely reassured that the patient could be sent home or reviewed again when an accurate reading would have indicated they should have been sent to hospital. And therefore, inaccuracy is a patient safety issue. And that's the point that I really want to finish on, that we are talking about not an academic situation where a small level of inaccuracy doesn't matter. It does matter. This is a patient safety issue, because a small level of inaccuracy can lead to a wrong assessment, wrong management of the patient, and a wrong clinical decision being made. And that results, as was found during the pandemic, in increased morbidity and increased mortality, especially when you are dealing with large numbers of patients. So if we are preparing for the next pandemic, we need a device that make sure that it is accurate in all patients, and even infrequent inaccuracies will be a major problem in a pandemic. And therefore, my take-home message to you in finishing is this, that if we want to improve patient safety, we have to improve pulse oximeter accuracy. And that is about making sure our equipment is accurate, and not just the equipment, but the maintenance and frequent calibration is accurate. We need to make sure that this equipment works in all groups of patients equitably. And we need to make sure that when we perform pulse oximetry in our patients, that we are performing it correctly to reduce the risk of inaccuracy. And what that means is that even with perfectly accurate pulse oximeters, we need to improve education as well as improving accuracy. We need to improve education so that doctors, nurses, paramedics, physician associates, everybody gets some training in pulse oximeters and pulse oximetry at undergraduate and postgraduate level, so they know how to interpret it and evaluate pulse oximetry readings in whatever specialty they're going to end up in. We need a CPD program to backfill the level of knowledge that is required in those people who've not had any training in pulse oximetry. We need a guideline, please. We need a guideline on pulse oximetry so that all the other NICE guidelines can be linked to it, so that people, when it says in a guideline, these are the readings you should be doing something with, there is an asterisk there that says go to the pulse oximetry guideline, which says it's important to think about accuracy. These are the factors. This is how to perform pulse oximetry. We need a guideline, please. And we need good resources, just like the respiratory surge program for children with bronchiolitis had an e-learning for health module. We need a pulse oximetry module that we can all use for our training, wherever you are and whoever you are. And so that is my take-home message. Over to you.
Thank you so much, Paul, for that excellent talk. So my name is Huzaifa Adamali. I'm a NHS respiratory consultant, a pulmonologist, which essentially means I spend a lot of time thinking about lungs, oxygen, and occasionally wondering why people who are breathless still insist on telling me their entire medical history in one long sentence. Today, we're going to talk about pulse oximeter, those tiny devices that have somehow gone from a specialized hospital equipment to something your neighbor bought on Amazon during COVID, just in case. My role today is to take you on a whistle-stop tour through what pulse oximeters do, how they can help, how they can mislead, and how they can prevent them from ruining an otherwise lovely clinical decision. We'll talk about guidelines, pitfalls, racial bias in readings, and what we can do to improve the way we use these deceptively simple gadgets. Think of today as 50 Shades of SpO2, educational, slightly provocative, and occasionally uncomfortable. But fear not. By the end, you will never look at a glowing red fingertip in the same way again. Next slide, please. So let's start with the basics. Pulse oximeters are little plastic clamps that look like they belong more in a manicure salon than an ICU. You pop them onto a finger, and like magic, it gives you a reading of oxygen saturation and pulse rate. But behind that magical glows lies that sophisticated physics. I'm full of with all the mechanical engineers that are in the audience. Light absorption wavelengths, arterial pulsations, and occasionally, if the patient just eaten crisps, pure confusion. In hospitals, pulse oximeters are everywhere. We see them in my outpatient clinic, in emergency departments, wards, theatres, ambulances, and even in the pockets of enthusiastic junior resident doctors, along with the 27 pens and no idea whose they are. They have become the fifth vital sign, the one number that everyone stares at. Family members are looking at the monitor like it's the stock market. Nurses try not to roll their eyes when someone says, but it was 92% at home, as if home saturation values follows them into hospitals like royalty points. The key message is pulse oximeters are incredibly useful, but only when we remember they are guidance tools, not mystical truth-telling devices. Ah, yes, and guidelines. The NHS national sport. We have guidelines on how to use oxygen, guidelines on when not to use oxygen, guidelines on whether the guidelines should be followed. For pulse oximeters, major bodies like NICE, SIGN, BTS, the Sepsis Trust, and the Royal Colleges all agree on one thing: use SpO2 values wisely and always in context of the patient you're treating. News 2, for instance, turns oxygen saturation into a kind of behavioral scoring system, like a Fitbit for illness. Drop below 92%, and suddenly everyone's heart rate, including the staff's, mysteriously increases. We have thresholds for asthma, COPD, pneumonia, bronchiolitis, COVID, and sepsis. These guidelines help us triage, escalate, de-escalate, and occasionally scratch your heads when the pulse oximeter insists a patient with no symptom is dying. Guidelines are essential, but they assume the device is telling the truth. The assumption here is optimistic. In asthma, pulse oximeters are like speedometers, great for judging severity, terrible if you are relying on them while ignoring everything else. In the emergency department, SpO2 gives a rapid sense of how severe an exacerbation is. A low saturation tells you to act fast. A high saturation might give you a moment of smug relief. Although asthma patients can look deceptively well right until they suddenly aren't. But the oximeter has its limitation. A moving child? Children are like caffeinated squirrels running around in your emergency department. Cold fingers turn the reading into a random number generator. A nail varnish, just forget it. And henna, well, henna, the dye you put on your hands, even worse, and not mentioned in most guidelines. And yet families still look at me like I have personally insulted their ancestors when the reading is different from the one on the home device bought for £4.69. Pulse oximeters are helpful, but in asthma, the patient's work of breathing, ability to speak, their respiratory rate, and gut feeling from the clinician are just as important as the number flashing on the screen. The oximeter is a tool, not a diagnosis. Next slide, please. Let's be fair. Pulse oximeters are brilliant. They help us catch silent hypoxia. They guide oxygen therapy. They save lives. But they also have a dark side, like Darth Vader, but with LEDs. The good stuff is they detect silent hypoxia, they're non-invasive, they're quick, they're cheap, and they don't require the painful arterial blood gas. Everyone, including the patient, should cheer at this. But the not so good stuff, it depends on the type of skin. It drops to zero if the patient flaps their hands like a pigeon. It freaks out with cold fingers. It's wildly inaccurate on cheap devices. It encourages dangerous overconfidence, and it has caused more "but doctor, my oxygen is fine" arguments than any other intervention. Some devices struggle with darker skin tones, a flaw that has been highlighted repeatedly but has not been fixed fast enough. Others struggle with the hands, poor perfusion movements. All of these make you think whether these devices actually belong to a pound shop. The point? A pulse oximeter is only as good as the clinicians interpreting it. Next slide, please. Now we get serious. COVID has taught us many things, including the fact that pulse oximeters may overestimate oxygen levels in people with brown or black skin. That meant silent hypoxia wasn't just silent, it was hidden. ICU patients in 2020 were found to have normal SpO2, while arterial blood gas revealed dangerously low oxygen levels. Governments responded, the FDA responded, even the UK government, usually slower than a patient climbing hospital stairs, commissioned an independent review. We now know pulse oximeters can systematically overread in darker skin pigmentations. This isn't a new problem. It just took a pandemic to get people to notice. And device accuracy widely varies between manufacturers. This is not merely a technical issue, it's a health equity issue. Next slide. And here is the part where I pretend that I have the power to change national policy. 3 simple recommendations. Better training and public awareness. People just need to understand. Not all pulse oximeters are created equal. Not every reading is accurate. Cold fingers are basically enemy combatants. Patient-specific considerations. Guidance should explicitly address skin pigmentation, perfusion state, movement, device types, and physiological context. We need saturation ranges that reflect reality, not wishful thinking. And finally, hospitals shouldn't buy pulse oximeters that cost less than a bread sandwich. It is used to guide critical decision. It should be validated properly across all skin tones. If we get this right, we improve safety, equity, and decision making. And if we don't, we'll keep discovering hidden hypoxia the hard way. And my final slide. To wrap up, pulse oximeters are wonderful tools, convenient, clever, and clinically invaluable. But they are imperfect, they can mislead, and they can leave a physician with a false sense of security. Our job is not to worship the number, but to understand that the patient who's sitting in front of us. If we improve device accuracy, we need to train clinicians better and acknowledge variability between patients. We can use pulse oximeters safely, equitably, and intelligently. So thank you for listening, and may all your future SpO2 readings be accurate, reliable, and achieved without removing nail varnish. Thank you.
What a fantastic presentation. On behalf of Sam, I'm going to now run his video recording of his slide deck, but he will hopefully be able to join us for a few Q&A points in a little while. So over to his video.
Hi all. I'd like to thank Nick and IMechE for hosting this event. Sorry I'm not presenting this live, but I have a 1-week-old and he takes priority. Today I'll be going over my experience with continuous monitoring of SpO2, mainly in highly acute patients in a remote setting. Most of you probably don't know me, so I'll give a brief background to understand where I'm coming from. I've been developing medical products with hardware, AI, and software for over 11 years. In that time, I worked in R&D for Space Labs Healthcare. I ran the biomed team at Current Health from pre-seed to acquisition for Best Buy, and I ran product development at Lifebill AI. All of these companies focused on continuous monitoring of acute patients. And during that time, I'd have 4 FDA-cleared algorithms, including SpO2. I found that SpO2 is probably the best predictive signal of seriously ill patients. I know others may have covered how SpO2 works, so I'll go over this briefly. SpO2 is a surrogate metric for the direct measurement of arterial oxygen saturation through blood gas analysis. And while it's correlative, there is a difference, and that's important to understand. SpO2 is generated by firing red and IR lights into the skin and measuring the absorption. From the different absorption characteristics, we calculate the ratio of red to IR. Depending on the amount of oxygen in the blood, red and IR get absorbed in different amounts, and this gives us a variance that allows us to measure SpO2. The R value gets converted into SpO2 through a calibration curve, which is made on regressing against previously captured pairs of R values and gold standard blood gas measurements. For example, an R value of 0.4 may be 100%, and an R value of 1.3 may be 70%. The validation criteria is fairly simple. Take a group of healthy volunteers and desaturate them down to 70 or 60% blood oxygen level. At fixed levels, so 100, 95, 90, 85, and so on, they draw blood and measure the blood gas and compare that to your SpO2 readings. All devices need an ARMS of less than 3% to meet the relevant standards. Now I'm going to go over some of the issues with how current SpO2 monitoring is done and why bad readings can occur in the wild. Firstly, the pulse style signal is very weak. This is the AC value. It kind of looks like your blood pressure waveform. It's about 0.5% of the signal or less in people with shock. This means interference through other light sources or motion that changes the sense of contact with the skin, which is frankly all motion, will destroy the signal, and there is no way to recover it, even with AI. When there is noise, the ratio tends to 1, as both signals have been saturated, and that's all they're measuring is that noise. So for most devices, this is an SpO2 of around 82 to 84%. So whenever I see clusters around those levels, I would always check the contact and how the patient's been moving. Now, I talked about the R values being used to generate SpO2. R values are not a direct one-to-one mapping to blood gas or SpO2. It's not like measuring an R to R ECG interval peak. There are irreducible errors in this method. There are variances, as these can be dependent on hardware, subject selection, and the gold standard used. Some patients can be real outliers, and I found that there is a large range of R values mapping onto 100% SpO2 in particular. Different SpO2 monitors will also have different accuracies for different patients. SpO2 monitors are all wrong within a degree of accuracy, so that 3% we were talking about. They won't all be wrong in the same way. A patient may be 95%, monitor 1 may measure 98%, and monitor 2 may measure 92%. These could be different just from having a different patient cohort when generating this calibration curve. Skin color is very important in developing SpO2 monitors because darker skin absorbs more light. I think this is one of the most talked about, but probably least understood problem in monitoring. Because of the darker skin absorbing more light, more power is needed to penetrate the skin. In medical device design, one of the core drivers is to minimize battery usage. The lower the battery usage, the longer between charges or replacing batteries. To help this, devices will detect the skin color and adapt the power consumption to meet it, boosting that light penetration. This also means battery life will drain faster for darker skinned people. However, All dark skin is not equal. Dark-skinned individuals can vary by 10 to 1,000 times more light absorption while visually looking similar. This means if you're not including the darker skin tones, devices may not have been designed to have enough power to penetrate the skin. Other things like tattoos or nail polish can also have this effect. I believe this is a major cause of poor accuracy on dark-skinned individuals, even if designed and tested using the Fitzpatrick scale properly. We used our devices both on dark-skinned African Americans and people in Africa and found Africans absorbed 100 to 1,000 times more light. So this really is not a trivial issue. Devices can also miss fast SpO2 drops. In order to get more consistent readings, device manufacturers will often average over 1 or 2 minute time windows. This smooths over small variances to make validation and often real-world use better. However, your SpO2 can drop 20 to 30% within 10 seconds and recover within 30 seconds, 15 minutes, or can never recover without intervention. These are usually caused by postural or respiratory problems, but I saw this happen regularly in highly acute patients. If you're averaging over long time periods, a drop down to 80% may never register below 95 on a monitor. You may never be aware of these situations, especially in a spot check environment. How you measure SpO2 also matters. When developing a monitor, we did a real-world observational study where nurses would take their own SpO2 values and compare them against our device. We found that when we gave low SpO2 values, corresponding nurse readings would be normal. We followed them to view what was going on, and we found that they would often wake or arouse the patient before the measurement, fundamentally changing their SpO2, and that our readings after the arousal would also agree with those nursing measurements. This is really important for spot checks, especially in remote settings, as it can give false impressions of the patient's SpO2. Drops are common overnight when patients are not being monitored and never get reflected in the data, especially if there are measurements being taken by an aroused patient in the morning or by a clinician. Lastly, please calibrate and make sure you have suitably performant monitors. A large study in NHS hospitals looking at SpO2 monitors found 10% of the monitors had electrical errors and a further 22% had spectral drifts. That's almost a third of all monitors being used. Both of these issues will cause false SpO2 readings. You need to make sure your devices are regularly calibrated and checked. You can't leave them on the shelf for 3 years or just buy one off Amazon and expect them to be calibrated. A further study out of UCSF looked at the most commonly available monitors and found 5 and 11 do not meet the 3% requirement for validation, even though they had previously passed a validation study. You need to be aware of this and don't take procurement lightly. I'd like to thank you all for listening, and if you have any more questions, please reach out to me at sam@genora.ai, or you can read my blog on my website for more information on the wider medtech ecosystem. Thanks again to Nick and IMechE.
Perfect. We've heard some amazing people, and I sort of come in to this as a bit of a trespasser, but having used pulse oximeters for some time. So what I'm going to try together is bring together some of the thoughts. We may not quite have quite as much time as we wanted for comments, but please do put those in the Q&A. So basically, what I want to really look at is the guidelines and how can our guidelines be safe when we're using a device that's where the accuracy is quite variable. An example of this, we have 12 National Institute for Clinical Excellence in the UK that have a cut-off measure. And are our oximeters that we're using actually accurate enough to be used in this way? Or could it be as reliable as tossing a coin? So just looking through the ISO standards, which have already been mentioned, the ones that we use for EU and MHRA give a plus or minus 4% accuracy tolerance. Now that's based on 10 healthy volunteers, or minimum of 10 healthy volunteers. And with that, there's actually no skin color specified in the ISO standard. I'll talk to you about the FDA in a second. A total of 200 data points and only giving you an average accuracy of plus or minus 4%. Now, I'll talk about the average in a second, but that means if you've got oxygen level shown on your pulse oximeter of 93%, is it not 89% or 97%? You just don't know. So if we could just break that down a little bit further by actually focusing on the FDA, which uses 3% rather than 4%, And again, they use 10 people, but their specification is 2 of those 10 have to have darker skin tones, but don't specify what. So it's not really statistically powered to show racial difference or skin color difference. But they say, quote, because this is an average accuracy, only 2/3 of the readings will fall within that 3% or 4% for Europe and ISO standards. 95% of those readings will fall within twice that, so 6%, plus or minus 6%, or 8% if you're using ISO standard. So in 1 in 20 values, it could be more than twice as accurate. That's more than 6% out, plus or minus 6%, or plus or minus 8%. So you can see these devices get through medical regulatory approval with a wide range that really crosses the span of healthy to unhealthy. If we look at the racial inequality or skin color inequality, which would also be framed, is this was known for 30 years, but really came out with this New England Journal of Medicine paper, letter in 2020, where they showed that people with black skin in an intensive care unit with COVID-19 were 3 times more likely not to have their low oxygen recognized than someone with white skin because of these devices. They looked at 10,000 patients in ICU with COVID-19. 13,000 of those measures had a low accuracy reading where basically someone's arterial line measurement was 88%, so the gold standard, but their pulse oximeter reading was shown 92 to 96%. And they broke this down into cohorts. And the better cohort, almost 12% of black people in ICU with COVID-19 had this unrecognized low oxygen level, and almost 4% of people with white skin. So there's an issue with not just skin color, but everyone, and it's multiplied with people with darker skin. And this is significant. 4 to 8% is a massive difference. So the FDA, as we heard, well, I don't think we did hear, but has come out suggested we get better standards, but we're still awaiting those. There'd be some suggestions that you should testing on 24, minimum 24 people across the skin tones, and there's something called a Monk skin tone score. And so that's likely, I believe, to be adopted. The UK independent government report talked about equity in medical devices, and almost 2/3 of that report was based on pulse oximeters. And this is to such an extent that in the US, multiple medical device manufacturers are being sued for racial inequality because of their pulse oximeters. So there's obviously a need to really resolve this rapidly. So some existing evidence has been quoted to suggest that there isn't a skin color issue. Some current large manufacturers have retrospectively reviewed their data, but they've pulled their data into light skin and darker skin. And as you say, this is a Massey scale, darker skin on this side. And there's quite a lot of overlap. And as you can see, there's only one subject in each of the darker skin tones. So I think we have to take this sort of argument with a large pinch of salt, and we really need statistically powered to show differences. There's also been an argument with similar manufacturers that actually there isn't a difference in the signal strength if you've got black skin. And in their article, they quoted if you took a cutoff at 1, there was the same number of people below 1 with white or black skin. However, if you take out the cutoff of 0.5, you see that almost everyone below 0.5, so a low signal, has black skin. So I think be really cautious when you're deciding your oximeter and the data you're looking at to decide which one. So University of California, San Francisco was briefly mentioned, has decided to start open oximetry program led by Professor Michael Lipnick, who was hoping to be on this, but because it's Thanksgiving, he's not, unfortunately. So hence I'm presenting. So the Open Oximetry project is looking at objectively measurement of finger pulse oximeters against a new standard, which is a new proposed SDA standard, which is 24 people across all skin tones, but comparing it with the 3% accuracy. Specifically set out to look at the skin color differences, but in doing so is actually highlighted the difference between the guidelines, the 3% or 4% FDA standards, and the actual achievement of these devices. So they've got a wonderful website. Have a look at it. You can actually choose by manufacturer. And if you choose by manufacturer, you get all your favorite oximeters come up. And it shows how they perform against this FDA standard. As I say, the FDA standard should be less than 3%. These are all devices that have passed FDA. And as you can see, these ones are all above 3%. And some of them quote a manufacturing quote of about 1.5%, but only achieve 3.48% in this testing. I must say this in this testing. The other thing is they break it down by skin color. So on the left-hand side, we have the lighter skin tones, middle is the middle skin tones, right-hand side, the darker skin tones. And at the bottom, you can see SaO2, which is the gold standard, the arterial oxygen, taken from your radial artery, which is the gold standard. And then the dotted line is where above that means that the finger oximetry is over-reporting. And as you say, that's quite worrying, because if you look across the whole skin color, almost the majority are being over-reported. And that means that people are being overly reassured. And I'll talk about things like this 8% out, so in a minute. But I think there's a worrying trend that we can see this, and this was a very high-end pulse oximeter that I've chosen. And as we said, these are assessed against arterial O2. And if you look at the ISO standard, it does talk about the arterial O2, and this is the quote, although I won't go into great detail. So Bland-Altman plot is this. In some situations, you plot other things, but if you've got a gold standard, you plot the gold standard at the bottom. So this is a similar manufacturer as we looked at before. They quote their accuracies as plus or minus 1.5%. As we know, the ISO standard suggests plotting against arterial O2 only, but in here they're plotted against the average of the arterial O2 and their performance. So if their device measured 96% in someone whose actual oxygen level was 90%, in here we'd have a difference of 6%. In here, because we're averaging x-axis to the bottom, we've only got 3%. So this may explain why in some situations we get a quote from the manufacturer of a very accurate device, but on objective testing, very open oximetry, in the limitations of any testing, it's showing higher. Open oximetry has got a preprint out looking at 34 of the units that they investigated. 29 of those were regulated medical devices. 6 out of 34 failed the ISO standard of plus or minus 4%, and all 6 of those had got CE marked from were regulated medical device. 12 out of 34 failed the FDA guidance of 3%. 8 out of 12 had passed FDA clearance. So we've got a big issue here in that even if you've got FDA cleared device, the accuracy root mean square is not necessarily how it performs in clinical practice. And remember that open oximetry is looking at healthy volunteers between the age of 22 and 55. And this isn't even real world performance. So we've looked at accuracy. We know that there's quite a variation. And we've talked about guidelines. And so guidelines throughout the UK are pretty ubiquitous, containing cutoffs. So there are 12 NICE guidelines for children, for pregnancy, for sickle cell infections, cardiac chest pain, neurological. The cutoffs also come for asthma. So this was the first time it moved out of hospital, was in the BTS sign guidance, moving pulse oximeters out of hospital into primary care. And they can decide how quickly you're treated and what sort of treatment you get with asthma. The same for Sepsis Trust, looking for sepsis, and News to Early Warning Score, which I'll talk about in a second, which is a UK-based score, but other countries will have different scores. So why is there a problem? I believe the problem is because these devices were developed for anesthesia and intensive care, where you're monitoring a trend. You've got the patient in front of you, and even on this supposedly stable patient, you can see there's a great variation. But if you've got someone in front of you, you've got multiple data points, you've got the patient as a reference, and you're making clinical action based on the trend and the clinical state of the patient. You're not just going by a number. However, if you just got one number, and it shouldn't read 89%, but it reads 8% out, 97%, I think you've got a problem. And I think that's where we've got a problem with clinical guidance at the moment. We're relying on point measures to give guidance. Now, guidance is guidance, but we know in medicine, particularly when you get less experienced staff, or in a situation in COVID, where it becomes very much treat the number, not the patient, whereas we'll always talk treat the patient, not the number, we get into problems. So if you look at the early warning score consists of things like blood pressure and pulse and breathing rate and temperature and oxygen levels. And basically, if someone's in hospital, it's used if someone's in hospital to look to see if they deteriorate, to see whether there's an escalation of care, to call the senior nurse, to call the doctor, to call the intensive care team to actually help someone out if they're deteriorating. And so you add up these numbers. So for oximetry, you get a 0 score for 96% or over, and a 3 score of less than or equal to 91%. And you add up these scores, and it decides how intensive your treatment and monitoring is. You can see if you get a down selection to a lower number, it actually dilutes your score. So this is particularly important if you look at the same high-end pulse oximeter. If we look at the 90 to 93% areas, and we actually take the offset that these pulse oximeters are actually reporting, You see, out of these 40 subjects, 23 of them would be miscategorized as over 93%, moving the risk upwards or downwards even to deflate the actual score and mean that there's less acceleration of care. That was in white skin or light skin. In people with darker skin, you can see only one of these readings in these subjects who are healthy volunteers successfully was reported between 90 and 93%. All the rest were reported as reassuring or above 93%. I think there's a big issue there. Just another way with warning scores to see how they work is if you've got a 1 score for one parameter of 3, that automatically puts you into a situation where you're looked at quicker. So if with oximetry, your level is under 91%, there's an immediate flag. So with people with lighter skins, 10 people were misclassified as over 3. That's 12%. And people with darker skin tones, more than 50% were miscategorized. So they didn't have the opportunity to earlier escalation of care because of their signal parameter reading. So there's a problem, whatever your skin color, I believe, but obviously that's multiplied if you've got darker skin tones. So we talked about accuracy of finger oximetry. We talked about how it affects cutoff. How do we mitigate this in clinical care and guidelines so that we can safely be treated? I raised this with a nice prioritization board with a formal white paper saying that they had 12 nice guidelines with cutoffs and what's the effects of accuracy on patient safety. That was formally considered in their prioritization board, and you can see on the website their report that they felt there was enough warning within their guidelines to resolve this, and they didn't feel that they needed to review this. I took issue with this. You had a chance after you got the response to go back with 3 questions. My 3 questions were based on, in their guidelines, they had a very soft warning that was hidden in the guidelines, wasn't on the crib sheets everyone has or the summaries. It says some pulse oximeters can underestimate or overestimate oxygen levels, especially if the saturation level is borderline. I think that's debatable. We've seen that we can see plus or minus 8% very easily, and we had those examples. Overestimation has been reported in people with dark skin. I think there needs to be stronger safety issues. So I went back to them and said, how did they consider the potential patient issues of the plus or minus 8%? And they said that they're not responsible for medical device regulation and safety, and passed that back to the MHRA. I said, how did they determine if the information they were giving in the warnings they were giving were being considered by clinicians? They said it was not responsible for them to assess the competency of clinicians. And finally, I said, how do they assess the possible risks of harm of their guidelines and why they're not investigating further? And they said there was absence of evidence of harm and they didn't know how to quantify that. For me, that's not good enough. Guidelines have to take in human factors. They have to take in how people read them, how people look at them, and ensure they're very easily read and any warnings are proportionate and not hidden in small print. So my takes are low SpO2, low oxygen level for a finger oximeter, take action. Yeah, there may be other reasons for it, but you need to know why. If it's normal, for me, it is not reassuring. We should not be using these reassuring devices. I believe for those who procure and use these, to inform your staff, check your oximeters about open oximetry. Also check that they actually are medical devices. They often have CE and UK CA mark just for electrical safety. They may not actually be a medical device. So I think we've said before, my feelings are we need to publicize this, which is what we're trying to do today. And I'm sorry if we don't have enough time for debate. We need to look at guideline review to change them to make them safe. We need the regulators to change standards, and we need manufacturers to step up, label the inaccuracy and improve the accuracy. So you've heard too much from me. You've heard about the use in primary care, hospital care, technology limitations, and how objective measures have shown that there are problems. So I'll stop there and ask for hands up or any questions, and perhaps Helen can help us with that. Thank you. I believe we may be able to see on the participant list. I don't think we have had hands up yet.
We've got a couple of questions actually, Nick, in the Q&A section.
Fantastic.
One with regard to what is your opinion to make a right validation of SpO2 against SaO2, and how can you do this in your hospital? I don't know if you want to take that one first. And there's also one, is there a calibration interval for pulse oximeters?
I think Sam, you've done a lot of calibration and validation, haven't you? And from what I understand, there's no actual recommended calibration once the device has been manufactured. But I'll pass on to you on that one, Sam.
Yeah, so I think there's two uses of the word calibration here. One's the calibration curve that maps these R values that I was talking about onto SpO2, so basically calibrating against different actual people's SpO2 readings. And then there's a second calibration, which is, are the wavelengths of the lights that you're using what they should be? So a lot of pulse oximeters, for example, can have drift in the actual physical manufacture of the device itself. So if you're meaning that, you can check that once a year with the manufacturer's tool. And if they're not accurate, then you shouldn't really be using that device. For calibrating in a hospital, the testing is actually quite aggressive. So if you're talking about desaturating people down to 70% oxygen, I've seen hyperfit people really struggling with that, physically shaking and doing things like that. That's why you can't get, people with COPD or serious issues to come to go to these studies, is because it is really aggressive and you do need to be set up for it. I think theoretically there's no issue in actually being able to do it, but having the right setup with people who are trained and understand physiological reactions and be able to do it properly. I think there's places like UCSF, for example, it's the main one, but there are a few others coming up.
Excellent. Thank you. Oh, there's another one just come in. Is there any understanding of a trend of deviation away from calibration over time?
Sam, I think you are best placed again, being the technologist on this.
Yeah, so I think there was a little discussion that I saw in the webinar chat about the numbers of people that are needed in the actual study, and the calibration in terms of SaO2 mapping can be really dependent on the group of people that you get. So it's not even just like a drift over time, as in, there's 5, 10 years, but there's also a fact that we saw drastic changes from cohort to cohort, and we had to do multiple studies in order to get a reasonable, facsimile of these R values to be able to be, across the spectrum. One of the other things I didn't touch on, but is really important to understand, is a lot of time medical device manufacturers will release newer generations of their systems and try and prove what's called functional equivalency to the FDA. So they say that, everything in the electronics is exactly the same, therefore we don't need to revalidate the system. I'm not sure if that's still going on. It's been a little while since I've been on that end of things. So you could have multiple generations of devices which are quote unquote functionally equivalent, but have never actually been revalidated, even though there's like a new housing or processor or things like that. So drift can come in that way as well. And then other than that, the lights can just come out of whack. So you buy one on Amazon and it gets shipped and it gets dinked around while it's in transit. That can definitely, impact things as well. So there's multiple ways that you can get inaccuracies and drift, if that makes sense.
There's another question coming. I think it's sort of along the same lines. What are your thoughts on accuracy, precision of BGA devices, cross labs? Is the last part of that.
Oh, is that for me? I don't know what BGA devices are. If you could elaborate, that'd be great.
Okay. Blood gas analysis.
That's not my end of the things. I just see that as the gold standard. So I've not done a blood gas analysis machine myself. I'd be really interested in it. That stuff's fascinating, but I've never done that.
I think I'm no expert. There may be other people on this call I've certainly seen, I think, quoted about plus minus 1% is I think the variation for a co-oximeter to be used, but I couldn't quote a specific, but there is obviously variation of your gold standard. So yeah, that's an equal problem as well. So your gold standard is never going to be 100% accurate. There's going to be some variation there as well.
And thanks, Paul, for adding in your link to your paper. And it's been really nice to see so many people leaving references to papers as well, which I think we'll try and collate together at some point. Looking at the time, if there are any other questions, that would be great. If not, I'd love to get a wrap up from each of our speakers, if that's possible.
I'll go last.
Okay.
So,
I'll
yeah.
go first then. So what I would say is that two things. Look at it from the clinician's point of view and look at it from the patient's point of view when it comes to primary care. From the clinician's point of view, we see pulse oximeters as being really valuable, really reliable, and really accurate. Okay, so if you put a pulse oximeter on the finger and it says 97%, you feel you're using it for reassurance. It's ruling out anything too serious. If that reading was to come back at 95%, you'd be feeling a bit more uncomfortable. If that reading came back at 94%, you'd be picking up the phone and ringing the nurse to bring in the oxygen and calling an ambulance. And therefore, if you are unaware that pulse oximeters can be inaccurate, that gives you a very false feeling of being able to say this patient is not big sick. So that's from the clinician's point of view. Clinicians need to know about this stuff, but they're not being taught it at medical school or anywhere else. From the patient's point of view, it is about patient safety. It is absolutely fundamental. Assessment, management, and decision making are being performed on the basis of absolute numbers or very narrow ranges of numbers in a device that is not that accurate by clinicians who are unaware that it is inaccurate and unaware of the factors that can cause it to be inaccurate and the consequences of it. And therefore, we need, Nick's talked about guidelines. We need a guideline more than anything. We need education, guidelines, and people need to be aware of this problem. Because as someone who teaches this almost every week, when I speak to clinicians, they are unaware of most of the things that we are all concerned about.
Thanks very much, Paul. Safer.
I think I've got four messages for all the being a clinician. I'm coming like Paul from a different angle. I feel that pulse oximeters are really fantastic tools. Just don't trust them like your favorite child. Trust them like the other one. That's my first. The second one, SpO2 is a guide. It's not a prophecy. Use your clinical brain, just not the blinking number. The third, I think pulse oximeters lie. Patients breathe. Always prioritize the thing with the lungs. And then the final one, I think we should treat pulse oximeters like a relationship. If something doesn't feel right, just don't ignore the red flags. Those are, I guess, the four things I would give to the audience. I hope a few clinicians are there listening.
I think they are very, very memorable points. Fantastic. Thank you. Sam, do you want to come in with the last point before we go back to Nick for a closeout?
Sure, I'll kind of go from what I've seen just in continuous monitoring, because it's not really been done with SpO2, especially in a home setting. There is a lot of information in the SpO2 signal that no one ever sees. Consistent fast drops overnight, especially with postural changes. In really sick patients, I would always see plenty of drops in SpO2 rapidly and then resaturated. You will be getting an inconsistent picture if you're just taking that one spot check in the morning. And that shouldn't reassure you about that patient that you think is really sick. And I know there aren't really, there aren't guidelines for this and there aren't too many options. Full disclosure, I used to work at a company that did remote patient monitoring, so this is where it's coming from. But from my personal opinion, there's such a rich vein of information to research on this. I know there's researchers in the call, so you should really start looking at it.
Thank you.
Fantastic. And for my part, first of all, can I thank everyone, particularly Helen, for putting this on, and everyone for support, and for everyone spending the time. And for those who did get out of bed on Thanksgiving, thank you very much, or are listening to that this afternoon. Fantastic. Um, for me, I was shocked 12 months ago when I realized the device I've been using for 14 years was so inaccurate. And every clinician I speak to, first of all, isn't aware, and second, is shocked. So I think a lot of this is knowledge. And I think the other thing is guidelines, although are guides, become embedded in culture. And if you've got a figure, a number on a guideline, you need to have in really big numbers as well next to it, the range that that number could be. Because otherwise people will take the number. You know, if it says to 93%, people will go oh, plus or minus a bit. But actually, if you say this 93% could be 89 or 97%, and it's in your guideline, in your crib sheets, then people will begin to realize, and they need to know the strength of that piece of the jigsaw. So diagnosis and treatment is all bits of jigsaws that you try and put together. And you've got a bit of a fluffy, woolly bit of jigsaw, which is your pulse oximeter. It isn't a precise, it's something that can give some information. So for me, they're valuable if it's low and you're not expecting it. If it's normal, from what I've shown you, I would not be reassured. So I think we need to ask those people writing guidelines to reconsider for patient safety issues. And I think we need better pulse oximetry if we're going to use it in these sort of ways. So I think that's my end. So Helen, I should pass on to you, but thank you.
Well, we're still getting a few questions in, but we're running out of time. So any questions that we've got left, we'll collate together. We'll try and get them into a response via an email or something like that. But I will certainly say that this, for me, has been quite an eye-opening conversation. Something that, as an engineer, makes me feel slightly itchy on the skin that I can't fix this immediately, and I think I might have to go and work with Nick and Sam to do some of this. But on behalf of the Biomedical Engineering Division at the institution, thank you so much for taking your time out of your busy schedules to come and tell us about this really vitally important piece of technology and the impact it is having on society. This is a big message that we need to pass on, not just to the wonderful clinicians and engineers listening today, but more broadly to wider society, so they understand when they come to see doctors and nurses that the information that they're passing on may not be as accurate as they think it might be. So thank you to all of you for taking part today, and thank you for everyone staying to listen and adding to the conversation in the chat. It's been really, really useful. Thanks very much.